Hello friends, welcome to CEC Live Lectures. Dear friends, today in this session we are going to continue further in our series on cell biology. Today we are going to talk on programmed cell death, and for the discussion on the topic we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Chemistry, Hindu College, University of Delhi. Dr. Rawat is a prolific professor, and through him we always try to discover new and newer things in zoology, as he is the professor of zoology, and through him we try and get maximum knowledge. Friends, if you wish to ask questions from Dr. Varunendra Singh Rawat on today's topic, then do call us through our toll-free number. Our toll-free number is 1-800-110-430. I repeat, our number is 1-800-110-430. Now I would like to welcome our guest, Dr. Varunendra Singh Rawat, once again. Hello sir, welcome to the lecture.
Thank you. So today I'll be talking about a very important aspect of life. Life, as we know, is continuation of basic metabolic processes in the organism. But every organism which is born is going to die. Apart from organism, the cells and tissues which constitute that particular organism, they also succumb to death. Now the death could be because of an injury which is not a programmed event, or it could be because of certain events in the cell which occur one after another sequentially, and that is termed as programmed cell death. This programmed cell death is given another name, which is apoptosis or apoptosis. Now how exactly this particular programmed cell death occurs and which cells are susceptible to this particular chain of events, I'll be talking about that today. So this particular lecture will basically be dealing with the processes of programmed cell death or processes of apoptosis, and basically there are two different pathways, intrinsic pathway and extrinsic pathway, which basically culminate in the death of the cell. And how exactly this particular chain of event occur, that I'll be discussing in this particular lecture. So all the multicellular organisms, they are made up of a number of cells, and these particular cells they play a specific role in the organism. So they grow, they differentiate, and they divide. But one important aspect is related to them, that after a specific period of days to which these particular cells are alive, after that they succumb, after that they die. So how does that happen, that I'll be discussing. Now the starting from the development of the embryo, once the fertilization of the ovum occurs by the sperm, then the embryo starts developing, and a number of cells are formed by the process of differentiation, by the process of basically division of cells, then differentiation. During this particular development also, a large number of cells die, and this death occurs by a specific chain of event which is caused by specific molecules present in those particular cells of the organism. Now the complex process of embryonic development, as I have already told, it involves cell proliferation, it involves differentiation, and it involves cell death. Now most of the cells in the multicellular organism, they die by a normal physiological process which is termed as apoptosis. And in adult organism also, there is continuous formation of cells by division of the parental cells, and daughter cells are formed, and there is constant cell renewal also, that is, new cells are getting formed and old cells are dying. But if there is a process if there is a abnormality in the process of cell death, then it might lead to a large number of problems like cancer and autoimmune diseases if these events are not taking part properly. Now as I have already told, that apoptosis or the programmed cell death, it differs from the accidental death, because in accidental death there is immediate injury or death of the cells, whereas in the apoptosis or the programmed cell death, specific chain of events will occur which will cause the death of the cell. Now this is a carefully regulated process, and it is responsible for maintaining a constant number of cells in different types of tissues in the organism. If this particular process was not there, the number of cells in a particular tissue might vary, they might increase to a large number or they might decrease. So this particular process helps in keeping the number of cells in check. Also it provides a defense mechanism to the organism by killing, by subjecting to death these cells which are infected by various dangerous organisms like bacteria or viruses. Now these particular cells which are infected by organisms like bacteria or by viruses, or which have been damaged due to some other processes, due to certain other metabolic events, they are subjected to apoptosis. Now this particular set of events, that is termed as apoptosis, plays a very important role during the development of the organism. Now if we consider larval development of amphibians, we know that there is a constant remodeling of the tissues of the larvae of the amphibian, and there is specific elimination of certain type of tissue which is remodeled to give rise to new type of organ. So this particular killing of the specific cells in the larva of the amphibians is carried on by the events of apoptosis. Also, there is an example, very good example of apoptosis which occurs in the embryonic development of mammals, is where number of neurons are getting formed. So the number of neurons which are getting formed in the embryo, they always exceed the actual number of neurons required in the adult organism. But why is there an extra set of neurons being formed? Even though the extra set is being formed, it is not continued in the adult organism, because only those neurons which are able to make contact with the target cells, they are able to survive, because these cells will then release certain growth factors which help the neurons which have made contact with them to sustain themselves. All the neurons which were not able to make contact with the target cells, they will die, because they will not be receiving any growth factors from the cell. Now all the neurons which are dying, how do they die? They die by specific set of events which we term as apoptosis, which I will be talking about in detail later on. Now the human embryo has about 50% of extra neurons which are going to be present in the embryo than which are required in the adult, and all these extra neurons are like they are subjected to death by this particular set of events. Now this particular set of events is highly regulated. It is not an irregulated process, and whenever that occurs, it gives certain morphology or certain specific physical changes to the cell which is being subjected to the apoptosis or programmed cell death. Now the cells which are undergoing this particular programmed cell death, they show shrinkage in their volume. That is, they are the overall volume of the cell. It reduces. The cell shrinks in volume, size, and also the nucleus shrinks in its volume. Then it also leads to a loss of adhesion of the cell to the neighboring cells. Most of the cells, except the RBCs and WBCs, which move in the different vessels of the body, blood vessels of the body, these cells are all of the cells are in intimate contact with the neighboring cells. Now whatever cells are going to get subjected to apoptosis, they lose contact, they lose adhesion with the neighboring molecules, and this loss of adhesion is very important factor. The adhesion of neighboring molecules is required for the cell to function normally. Now the cells which are going to die, they are losing their contact or adhesion to the neighboring molecule. Also, once the process starts, a number of projections or blebs start getting formed on the surface of the cell which is subjected to apoptosis, and the chromosomes start the chromosome of the cells, they are start breaking into fragments. Once the process has almost culminated, the cell breaks into small apoptotic bodies which will then be phagocytosed. Now these cells which are subjected to death by the process of apoptosis, they break into small apoptotic bodies, and then they are phagocytosed by macrophages. And these particular apoptotic bodies, how they come to be recognized by the macrophages? How do macrophages know that these are the cells which are going to die and which we are going to engulf these particular bodies? There are present certain specific signals which are recognized by receptors on the macrophages of the organism. Now these receptors might vary in type and number, but one such example of such a molecule which is presented on the surface of a dying cell is phosphatidyl serine. Now this particular phospholipid is present in the membrane of the cells of the organism. Now as we know that plasma membrane is a bilipid layer structure, this phosphatidyl serine is basically restricted to the inner leaflet of the plasma membrane. But the cells which are going to die, which are getting subjected to the process of apoptosis, they will be having phosphatidyl serine on the surface, on the outer leaflet, not on the inner leaflet. That means the phosphatidyl serine, which is basically present in the inner leaflet, that is transported to the outer leaflet so that it is recognized by receptors on the surface of macrophages, which then go and bind to these apoptotic bodies and engulf these apoptotic bodies. Now this is very important, because if a cell dies because of injury, then it releases all the contents outside, and once that happens, the immune system comes into play, and the cells of the immune system will start an immune response, which will lead to inflammation in the organism and pain, etc. But when this process of programmed cell death occurs, there is no release of contents of the cell outside, and the macrophages ingest the entire apoptotic bodies, which are formed by fragmentation of the normal cells. Now this particular term apoptosis, this was coined in the year 1972 by John Kerr and Andrew Willey and A.R. Curry of the University of Aberdeen in Scotland. And this particular process, the molecular basis, how which different molecules are taking part in apoptosis, this was elucidated first of all in the nematode C. elegans, that is Caenorhabditis elegans, and this occurred because of a fixed number of cells which are present in this particular organism. As we know that there are 1090 cells which are formed during the development of this particular worm, but out of them 131 cells are destined to die by apoptosis. So 1090 cells which are going to form during embryonic development, out of them 131 are sure to die, and the adult will be having 131 cells less than the 1090 cells. If these cells can be carefully monitored, and the process can be studied, how these cells are dying. Different molecules can be studied which are taking part in this particular process. The expression of different genes and their product and the activity of their product can be tested on these particular organisms, and this was done. Now it was found these particular strains, these particular worms were subjected to different types of mutagens, and it was found that certain strains of these worms, in those particular worms, after the subjection of these worms to mutagen, the death of those 131 cells did not occur. Now how does that happen, and which particular protein or gene was responsible for that? So studies were done, and it was found that a gene named as CED3 was responsible for that. That means the strains which were not showing death of those particular cells, they had mutated CED3 gene, so that they will be having a mutated CED3 protein product. More studies were carried out, and it was seen that there was a second gene which prevented programmed cell death, and it could be identified easily, and it was termed as CED4 gene. After that, studies were continued, and a new gene was found, CED9, and this particular gene, it was later on found out that it was related to BCL2 oncogene, and it inhibits apoptosis. So different set of genes were found which were promoting either apoptosis or which were inhibiting apoptosis. Like CED3 was promoting apoptosis, cell death of the specific type, and CED9 was inhibiting apoptosis. Now if CED3 was mutated, that means it was not able to carry out its function, then it cannot kill those cells, and therefore those cells were living in those particular strains. Now the pathway was worked out, and it was found that CED4, the second set of gene which was discovered, it stimulated CED3, and CED9 inhibited CED4. The CED4 has a mammalian homologue. CED4 is present in nematode, and this particular gene has a homologue in mammals which is termed as APAF1, and this particular APAF1 is able to bind to caspases, about which I will be talking later on, and it is able to activate them. Now a family of genes which are homologous to CED3 genes in the nematode has been found in mammals, and this family of genes is termed as caspases. Now these caspases are the enzymes or proteins which are responsible for carrying out the series of reactions which lead to death of the organism of the cell in a programmed manner, in a controlled manner or regulated manner. Now what are these caspases? These caspases are basically a set of proteins, a family of proteins, cysteine proteases. They are, because they are termed cysteine proteases because they have a cysteine molecule in their active site, and this is a family of cysteine proteases which is activated at an early stage of apoptosis and is responsible for triggering most of the changes which occur during the cell death. Now these caspases, they are very specific, as all enzymes are, and they cleave a certain specific select group of protein. and these caspases basically they may cut in the protein after an aspartate residue. That is the peculiarity or specificity of caspases, that they will cleave protein but only after the aspartate residue present in the target site. Now this set of family, family of caspases, they can cleave about 100 different target proteins in the organism in which they are found. In C. elegans, only one type of caspase is found, which is CED3, but in Drosophila and mammals, about 7 different types of caspases can be found. These caspases, as is obvious, they will be synthesized as inactive precursors, because if they are synthesized as active enzyme, then they will lead to death of all the cells which they encounter. But they are synthesized as inactive precursors, which then are activated by proteolytic cleavage, which then carry out activation of other caspases, and that's how different set of caspases get activated until the death of the cell occurs. These caspases can also be regulated in the organism by a set of proteins which are termed as IAP proteins, or inhibitors of apoptosis. IAPs inhibit caspase activity or they target these caspases for ubiquitinization and degradation into the proteasome. So they can either inhibit the activity of caspases or they can cause them to get degraded. These IAPs are present both in Drosophila and mammals. In Drosophila they are very active, because initiator caspases, which are almost chronically active in the Drosophila, they have to be kept in check, and this check is done by IAPs or inhibitors of apoptosis proteins. Now, what are the different targets of caspases? That is very important to know. These caspases, they are not focused on a specific set of protein. They have a wide variety of targets, like focal adhesion kinase protein, PKB protein, PKC protein, and RAF1 protein. Now, as the name suggests, focal adhesion kinase, inactivation of focal adhesion kinase is able to disrupt the adhesion of one cell to another. That means cells do not make contact with each other if the focal adhesion kinase is inactivated. This can be done by caspase. So the apoptotic cells are very easy, they easily release away from their neighbors. Then there are other kinases like PKB, which are able to disrupt the pro-survival signaling pathway, and caspases are also able to disrupt the generation of survival signals if they inactivate NF-kappa B pathway. So this NF-kappa B pathway is necessary for survival of the cell, but if caspases are able to disrupt that particular pathway by acting on specific proteins, then the cell will be subjected to death. Now, there are various targets apart from these proteins, kinases I have mentioned earlier. There are lamins which make up the inner lining of the nuclear membrane, and once the lamins are subjected to degradation or denaturation or by the activity of caspase, then it will lead to cleavage of lamins, which will lead to disassembly of nuclear lamina and shrinkage of the nucleus. Then there are proteins of cytoskeleton like intermediate filaments, actin and tubulin, which can be acted upon by the caspases, and their cleavage and inactivation will lead to change in the cell shape, which is required step for apoptosis. Then there are proteins like caspase activated DNAs, which are activated by caspases by inhibiting cleaving an inhibitory protein component from them, and once these proteins are activated, they will go, because they are DNases, they will go and cleave the DNA present in the cell, and this will lead to cleaving of the DNA of the cells. Now, the pathway which is followed by this particular apoptotic mechanism is, there are two types, basically extrinsic pathway and intrinsic pathway. Intrinsic pathway basically depends on the internal stimuli, whereas external pathway depends on the external stimuli which comes to the cell. Now, there occurs a crosstalk between the intrinsic and extrinsic pathway, but the end result is that the cell dies. Now, this particular panel shows that necrosis occurs without any regulation, whereas apoptosis occurs with regulation, and there are two types of pathway which are followed, extrinsic pathway and intrinsic pathway. Both require presence of caspase. Caspases are initiated, and this leads to execution, that is death of the cell. Again, it is carried out by another set of caspases. Now, what exactly happens in intrinsic pathway apoptosis? There are certain internal stimuli like hypoxia, damage to genetic component, or high concentration of calcium ions, or viral infection, or stress to endoplasmic Coulomb or oxidative stress, which triggers the apoptosis by intrinsic pathway. Now this intrinsic pathway, its activation is regulated by member of BCL2 family of protein. BCL2 family of protein is important in this context. These particular family of proteins are characterized by presence of one or more than one BH domains, which are basically BCL2 homology domains. So BCL2 family is important for carrying out the apoptosis through the intrinsic pathway. Now these BCL2 family members can be divided into three classes: those which are pro-apoptotic and those which are anti-apoptotic. Now pro-apoptotic members are those which basically enhance the apoptosis, and they have a large number of BH domains, and example are BAX and BAC. Whereas anti-apoptotic members are those which inhibit the apoptosis and they protect cell from apoptosis, and the example are BCL2, BCLXL and BCLW, and they of course also carry a large number of BH domains. Then there is another class which is BH3, which has only one BH domain, and it also promotes apoptosis but by an indirect mechanism. Now BH3 only proteins, example of which are BID, BAT, PUMA and BIM, they can exert their effect in two different ways depending on the particular proteins involved. They can either promote apoptosis by inhibiting anti-apoptotic members of BCL2 family, or they can promote apoptosis by activating pro-apoptotic members of the BCL2 family like BAX and BAC. So they can either inhibit the anti-apoptotic member or they can promote the pro-apoptotic members. Now these proteins are likely determinants as to which determine whether the cell will be subjected to death or survival. Now in the healthy cell, the BH3 proteins are not produced to a great extent, or they are not produced at all, but whenever there is cell is subjected to specific condition, then there is production of BH3 only protein, and they therefore shift the balance towards the death or survival of the cell. Now whenever there is production of BH3 only proteins, the restraining effect of anti-apoptotic BCL2 proteins are overridden, and this lead to production of the pro-apoptotic proteins like BAX, and these particular BAX protein, then they go and bind to the outer mitochondrial membrane. Once these BAX or BAC molecules are bound to the outer mitochondrial membrane, they cause formation of a channel in this particular membrane, which increases the permeability of the membrane, and this leads to release of cytochrome C into to the outside, to the intermembrane space, to the outside from the intermembrane space. Now release of this particular molecule is very important, and it's like a commitment step for the cell death, and there cannot be the cell cannot go back after cytochrome C has been released from the mitochondria. This is the point of no return, and once that happens, cytochrome C interacts with APAF1, ATP, DATP and pro-caspase 9 to form a multi-protein complex termed as apoptosome. These pro-caspase 9 molecule, as soon as they come in contact with this particular complex, they are activated, and this acts as this particular caspase acts as initiator caspase, which activate the downstream executioner caspases, which lead to the apoptosis of the cell. And there are of course other molecules also which are released from the mitochondria, like SMAC Diablo, and there is another molecule which is AIF, which is a flavoprotein, which also is like it's included in the apoptotic pathway, and it causes partial DNA fragmentation and chromatin condensation. Now another important gene, which is P53, is a tumor suppressor gene. It also plays a role in the regulation of cell death in many systems. Whenever there is damage to DNA, it leads to activation of P53, which promotes apoptosis through a direct activation of BAX molecule, or if there is an overexpression of P53, then the activation of BAX molecule is not required. Now P53 has been demonstrated to induce the BH3 family members like Noxa and Puma, and it demonstrates that there are multiple pathway of regulation of BCL2 family. Now this particular panel shows how intrinsic pathway of apoptosis works. There is damage to the cell, cytochrome C is released from the mitochondria, and apoptosome is formed, and this leads to activation of the initiator caspase, which leads to the activation of executioner caspase, finally leading to the cell death, which is termed as apoptosis. Now there is another pathway which is termed as extrinsic pathway, which differs from the intrinsic pathway because a signal comes from outside, not the inside of the cell, and the molecule which are responsible for that, like the extracellular messenger protein responsible for that, is tumor necrosis factor, which goes by the acronym TNF. Now this TNF is a trimeric protein which is produced by certain specific cells of immune system whenever they are subjected to adverse condition like exposure to ionizing radiation, elevated temperature, viral infection or toxic chemical agents. Now this TNF evokes its response by binding to a transmembrane receptor, and there is a large family of these receptors, the best known members of which are FAS and TNFR1. Now this trimeric TNFR1 protein is a member of family of related death receptors. They are termed as death receptors because they turn on the apoptotic process. These particular receptors have a cytoplasmic domain which contains a segment of specific 70 amino acids which are called death domain, and it mediates interaction between proteins. Now binding of the tumor necrosis factor to this receptor produces a change in the conformation of the death domain which leads to recruitment of a large number of proteins. The last protein to join the this particular complex is basically a pro-caspase 8 molecule. It is termed as pro-caspase because it is a precursor of caspase. Now this pro-caspase molecule, after it has gone it has been processed, they are held in close proximity with each other. Two pro-caspases are held in close proximity with each other, and they are able to cleave or process the other molecule. After this processing is done, a heterodimer is formed for each molecule, and after that a tetramer is formed, which is the activated form of this particular caspase, which is caspase 8. Now this caspase 8 is an initiator caspase. In the intrinsic pathway, caspase 9 was the initiator caspase, and this particular pathway, caspase 8, is the initiator caspase, and it initiates the cleaving and activating downstream or the executioner caspases which carry out the self destruction of the cells. Now this particular panel shows how that occurs. There is a specific binding of TNF to TNFR1, and that leads to activation of pro-caspase 8 by formation of a tetramer, and this pro-caspase 8 then activates is then activated, which then activates executioner pro-caspases, and then it leads to apoptosis or death of the other cells of the specific cell. So there are a large number of signaling molecules which are involved in this particular process, like FAS-L and TNF-F I have already discussed, and basic activity. The two pathways are basically they cross talk with each other. Intrinsic pathway and extrinsic pathway, they come together at a specific point, that is the executioner caspases, wherein the executioner caspases start the killing of the cell or they start the programmed death of the cell. That's all.
With this note, thank you, sir. Thank you so very much for giving us this session. Friends, we will be back after a short break and would be discussing more. Till then, keep watching us. Thank you. Hello friends, welcome back to this session. Friends, as you know that in our previous session we discussed on programmed cell death. Now in this session we are going to talk on cancer, and for the discussion of the topic we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is assistant professor and he is teaching in Department of Zoology. Hindu College, University of Delhi. Friends, if you wish to ask questions from Dr. Baranendra Singh Rawat, do call us through our toll-free number. Our number is 1800110430. I repeat, our number is 1800110430. Now, I would like to welcome our guest, Dr. Rawat, once again. Hello, sir. Welcome to the lecture.
So, friends, I'll be talking about a very important aspect of the cells, of the life in this particular topic, in this particular lecture. Earlier I discussed about the programmed cell death, and now I'll be talking about cancer, which is basically opposite of what exactly apoptosis or programmed cell death is. Cancer is basically unregulated growth and division of cells in an organism. We all have come across with the term cancer in our daily lives when we hear of people dying of cancer, young people dying of cancer, and it is basically a malady which has not been like no remedy has been discovered for this particular ailment as such. So what exactly happens? Why exactly cells start dividing uncontrollably? Why there is no regulation of cell division? And why does cancer occur in the organism? I'll be talking about that in this particular lecture. So I'll be discussing what exactly cancer is. To which type of cells do we give the title, or do we say that these cells are cancerous cells? What exactly are the causes of cancer? How does cancer occur? And what are the properties of cancer cell? How are we able to discern that these particular cells are cancerous or they are normal? And what are different types of tumors which can be found in body? What is the molecular basis of cancers? Which genes are responsible for causing cancers in the cell? And which different types of virus are responsible for causing cancers in humans or other organisms, that I'll be discussing. So the learning objective for this particular lecture would be that we should be able to tell about what are the different causes of cancer, what are the properties of cancer cells, and how they come into existence, how exactly cancerous cells develop in the body. So coming to definition of cancer, what exactly cancer is? It's basically a disease in which there is unregulated division of cells, that is abnormal cells are produced which are dividing uncontrollably. There is no control in the division of cells. In a normal individuals, in the different organs, the cells divide in a specific programmed manner at a specific time interval. But in cancer, there is no such control on the division of the cells. Now, how does cancerous cell come into existence? It is not an overnight occurrence. It basically occurs when there are a series of molecular events occur, which alter the fundamental properties of the cell. Now, what are those fundamental properties? I will be talking about later on. Now, what exactly is happening in a cancerous cell? The normal control system, which prevent the overgrowth of the cell, they are going berserk. That means the cells are not getting controlled by anything after there is a change, after there is a mutation in the cell, and after a cell has become cancerous, it might go and invade other organs of the organism. Now, these altered cells, those which are having certain mutations, they are able to divide and grow in the presence of signals which are normally inhibiting cell growth. So they do not require specific signal. That means a normal cell, whenever we subject that to a specific inhibiting signal, they will not divide, they will not grow. But cancerous cells are able to overcome that particular inhibition. Also, they do not require specific signal from outside for cell growth and division. They are sometimes cancerous cells are able to produce within themselves the signals which are required for growth and division. Now, as these cells grow, they develop new characteristics which are very much different from the normal cell characteristic. They change in the structure. There is a change in the structure of the cell. There is decreased adhesion with the neighboring cells, and there is production of new enzymes. Now, all these events lead cause a cell to become cancerous. Now, these changes are heritable. That means once that particular cell divides, whatever progeny it generates, it will also be having that particular changes, and that particular progeny will also be dividing uncontrollably. And that particular cell which is getting formed from the parent cell which is abnormal, it will be able to grow or divide in the presence of normal cells also. Whereas in normal cells, whenever they are in contact with other cells, their growth and division is inhibited. Now these particular set of changes, they allow the cancerous cells to grow and spread and invade the entire organs of the organism. Now these abnormality, how do they come into existence? Why does a normal cell become abnormal? Now these abnormal cells are generated because of mutation in specific genes of the organism. These protein coding genes, they get mutated, and whenever they get mutated, they lead to formation of a faulty protein. Now that particular faulty protein, if that protein was involved in the regulation of cell division, after mutation it's not able to control the cell division properly, and that is how these cancerous cells will come into existence. Now there are a large number of proteins. There is not a single protein. It's not that there is a single protein which controls cell division. There is a large set of proteins which is basically involved in regulation of the cell division cycle. Any of these proteins which gets mutated can lead to a change in the normal cell, and it might become abnormal, which does not have any control or regulation of its cell division. Now over the time, once a mutation occurs in that particular cell, over a time it is seen that more and more genes get mutated. Now this often occurs because the genes that make the proteins which are responsible for repairing DNA damage, they themselves are not functioning normally, because they also get mutated. So over a period of time, in that particular cells wherein has arisen a specific mutation, in that particular cell a large number of mutation follow, and as the number of mutation begin to increase in that particular cell, the abnormalities increase, and these abnormalities are passed on to the daughter cells. Now some of these cells may die. That means not all the mutated cells are able to survive. Some of them may die, but there might be certain changes or alteration in these abnormal cells which might be able to give selective advantages to those cells, so that they are able to multiply much more rapidly than the normal cell. Now we are saying that mutated cells might die, or it might have an advantage of over the normal cells, so that it is able to multiply at a faster rate as compared to the normal cells. And if that happens, that means it will lead to development of a tumor in the organism. Now this particular growth pattern describes most of the most of the mutant cells, which is way different from how a normal cell behaves in the organism. Now if a cell has accumulated a large number of mutations, then it is able to divide abnormally, and when it happens, then a large number of progeny cells will be formed, and these progeny cells will share the characteristic with the mutant cancerous cell. And once that happens, a tumor is formed. Now tumor is basically an abnormal collection of abnormal cells, and as long as the cells which are getting formed by division of the abnormal cells, as long as they stay in a particular place, as long as they are not moving from their location to other location, then that particular tumor is termed benign. That means it is not harmful to that particular organism. But if those cells gain a characteristic of moving, or they start invading other organs within the body, then that particular tumor is termed as malignant, and that is very harmful to the organism and also very difficult to treat, because if the cells, these cancerous cells, if they have moved from original location to a large number of other locations in the body, then the treatment will be very difficult. So the tumor from where the cells are moving outside to other location, that is termed as malignant. Tumor from which the cells are not moving outside, they are termed as benign tumors. Now the process of movement of cells from the malignant tumor to other locations in the body is termed as metastasis. So that means the cells are metastasizing from their original location and moving to different parts of the body, leading to formation of new tumors. Now what exactly is a tumor? As we know that cancer cells are behaving as an independent cells, they are growing without control, and this lead to formation of tumors in a series of steps. The first step in the formation of tumor is the tumor initiation, wherein there is abnormal proliferation of a single cell. That means a single cell which has acquired a mutation, there it keeps on dividing, and it leads to formation of a large number of progeny of that particular cell. This particular process is followed by hyperplasia, where there are too many cells which result from uncontrolled cell division. Now these cells appear normal, but changes have occurred in these cells which have resulted in some loss of control of growth. After that, the second step is, after hyperplasia, is the second step, which is termed as dysplasia, which results from further growth accompanied by more abnormal changes to the cells. And how do these abnormal changes occur? They occur by further mutation occurring in those cells. Now, the progression of tumor after the dysplasia event, the progression of tumor requires additional changes, and these additional changes can only occur when there are additional mutation occurring in the cell, which are leading to formation of cells which are even more abnormal than the starting cell. And after this occurs, there is a clonal selection. That means the cells which have accumulated a large number of mutation, they divide rapidly, and the clones of that particular cells, they become malignant, and they become dominant in that particular tissue. That means that tissue has a large number of those particular cells which have a large number of mutation. Now, these cells start to lose their original function, and these are termed as anaplastic. And after this happens, there might occur a stage wherein the cells will start moving from its original location. Till this particular stage, when the cells are anaplastic, they are not considered malignant. But if the cells acquire a mutation which causes it to move from that particular location to other location, they become malignant, and that's when the problem starts. Now, what are the different types of tumors which can be found in the body? Depending on the location, depending on the tissue which undergoes mutation, there is a class of tumors which have been described in the organism, in mammals specifically, and 5 classes of tumors have been described, and these are basically carcinomas. Carcinomas are the most prevalent types of tumors, which constitute the largest types of cancer found in the mammals. Now, carcinomas, they result from altered epithelial cells. That means carcinomas are tumors of epithelial cells. These epithelial cells which cover our skin and a large number of internal organs, whenever there is a change in their structure, whenever there is mutation in these cells, it can lead to formation of carcinomas. Then there are sarcomas. Sarcomas are other types of tumors which result whenever there is mutation in muscle tissue, bone tissue, fat tissue, or connective tissue. So, carcinomas are tumors of epithelial cells, sarcomas are tumors of bones, muscles, and fats and connective tissue. Then the third class is basically leukemia. Leukemia, it results whenever there is aberration in the blood forming cells, and lymphoma is a cancer of the cells of the immune system. Then we have myelomas, which are cancers of specialized white blood cells which make antibodies. So, the 5 tumors can be summed up as carcinomas, tumors of epithelial cells, sarcomas, tumors of soft tissues like muscles, bone, and connective tissue. Then leukemia is the tumors of blood cells, and we have lymphomas, tumors of the cells of the immune system, and myelomas are the tumors of the WBCs which make antibodies. These are the 5 classes of tumors which can be encountered in the body of humans, and of these the carcinomas are the most prevalent, making about 90% of the tumors in the body. Now, as we have discussed that cancerous cells, they become abnormal by accumulating certain types of mutations. And when they acquire mutation, they start differing from their normal counterpart. Now, whenever there is difference from the normal counterpart, there might be certain properties by which they differ from the normal cells, and there are a host of such properties which differentiate these cancerous cells from the normal cells. What are those properties exactly? Now, the first property is that they do not display density dependent inhibition of cell proliferation. Whenever we talk about normal cells in culture or in the body of the organism, specifically in culture, whenever they divide, they stop dividing after a specific density of the cell has been attained, but not so with the cancerous cells. Cancerous cells will keep on dividing even if the density of the cells become very large. So that means there is no inhibition of the cancerous cells based on the density of the cells present in the medium. Now, these particular cells also have reduced requirement for the extracellular growth factors. That is, they are capable of autocrine growth stimulation. Whenever we are talking of normal cells, they always require growth factors for their sustenance, for their survival, and their division. But when we talk of cancerous cells, they do not require any such input from outside. They are able to generate generate molecules which stimulate their own growth, that is autocrine growth stimulation. Then this cancerous properties might result because of unregulated activity of growth factor receptor. That is, it might happen that the growth factor receptors which are present on these cancerous cells, they might be mutated. So that means they are always active. In a normal cells, growth factor receptors receive the growth factor and then only they become active, which cause the growth of the cell. But in a cancerous cell, there might be mutation in the growth factor receptor, so that it is always active and it is always transducing signals which lead to continual growth and division of the cell. The next characteristic, which is also very important, is that these cells are less adhesive, and this particular characteristic is responsible for metastasis. Normal cells are very much adhered to their neighboring cells, but the cancerous cells, they are not able to adhere properly to the neighboring cells. That means they have less connection with the neighboring cells, and this happens because there might be mutation in there is less production of the adhesion molecule. The adhesion molecules which keep cells in close proximity with each other, they are less in number in the cancerous cells, and therefore these cancerous cells, they can easily move from their place, from the tumor to different place by the process of metastasis. Another property which is seen in the cancerous cell is that they do not show contact inhibition. A normal cell, whenever it comes in contact with other normal cell, it will not be divided further. It will be staying as such. But the cancerous cell, it does not show any inhibition even if it comes in contact with a neighboring cell. Cancerous cells, whenever tumor occurs in the body of the organism, the large number of cells are formed, and these large number of cells can only be sustained if they are provided with adequate nutrients, and the nutrients in the organism are provided by blood vessels. So cancerous cells, they produce certain molecules which promote angiogenesis. That means they promote formation of blood vessels in the tumor, so that these blood vessels, they will bring nutrients to the newly formed cells. So that is one more characteristic of the cancerous cell. Also, they are not able to differentiate normally. A cell which is formed, it is able to normal cell is able to differentiate normally, so that it can form its end product or the end cell. But cancerous cells, they are not able to differentiate, so they are not able to form the end product or the end cell in the organism. Also, many of the cancer cells, they are unable to undergo programmed cell death. So that is very important factor. Earlier we talked about programmed cell death. Many of the cancer cells, they are not able to be subjected to programmed cell death because of intervention or because of mutation in the pathway in the molecules which are responsible for the apoptotic pathway. Now, as we have discussed the properties of cancer cells, we must also know what exactly is bringing about these changes in the cancer cells, what causes mutation or what causes cancerous cells to be formed. A large number of causes are responsible for turning a normal cell into a cancerous cell. Among them are carcinogens. Whenever individuals are exposed to carcinogens, these carcinogens are chemicals which can induce malignancy in the normal cell. That means it can induce mutation in the normal gene. And apart from carcinogens, there are large number of agents like radiation, large number of chemicals and smoking which induce mutation in the normal cell, causing them to turn cancerous. Then there are hormones, proliferating agents like estrogen hormone, which is a proliferating hormone, which might also lead to a specific type of cancer like breast cancer in females. And this is a normal hormone being produced by the female, but it can also lead to abnormal proliferation of the cells. Then there are viruses which can cause cancer of specific organs like liver and cervix of the human uterus. And then there are bacteria like Helicobacter pylori which can cause ulcers, stomach ulcer in different organisms. So these are various causes which can lead which can convert a normal cell into a cancerous cell. Now, coming to molecular basis of cancer, these are causes, we have discussed causes, properties, but what exactly happens at the molecular level? What is the molecular basis of occurrence of cancer? Now, we have come to the knowledge that there are a large number of genes in the human genome, but only a small number of genes are responsible for causing cancer in the human. An alteration of these normal gene sequence can lead to development of cancer. Now, these malfunctioning genes have been broadly classified into three classes or three groups: proto-oncogenes and tumor suppressor genes and DNA repair genes, which are responsible for cancer formation in humans. Now, proto-oncogenes, they are the genes which produce protein products which normally enhance cell division or inhibit normal cell death. So, they are either enhancing cell division or inhibiting cell death. That is the function of proto-oncogenes. But when these proto-oncogenes get mutated, they form oncogenes, and these oncogenes are responsible for formation of for development of tumors in the body of the organism. Then we have tumor suppressor genes, which basically make protein that normally prevent cell division, or they produce protein which cause cell death. And then we have DNA repair genes, which help to prevent mutation, which can lead to cancer. Coming to oncogenes, the proto-oncogenes are basically the ones which code for protein which send a signal to nucleus to stimulate cell division. Now, they act in a series of steps which is called signal transduction cascade or pathway. Now, this pathway includes a receptor which receives a signal which activates a series of enzymes which causes the cell to divide. But oncogenes, which are formed as a result of mutation of proto-oncogenes, they are altered version of these particular genes, and these oncogenes activate the signaling cascade continuously. Whereas the proto-oncogene, they are producing proteins which activate or regulate the pathway at specific time. But once oncogenes are formed by mutation of proto-oncogenes, the product which are formed, they will be activating this pathway continuously, which is leading to uncontrolled cell growth and division of these cells. One example of such proto-oncogene is MYC, which codes for a transcription factor. Once there is mutation in this particular proto-oncogene, it will be coding for transcription factor continuously, and this will lead to uncontrolled growth of the cell. Then there is another family, which is RAS family, of which there are a large number of members, which function as an on-off switch in a signal cascade. Now, this particular family of genes encode guanine binding protein, which function in transduction of mitogenic signals from a number of growth receptors. But once there is mutation in the RAS, it remains in on position always. That is, it always remains in GTP bound conformation, which leads to uncontrolled cell growth. Now, about 30% of tumors like lung cancer, colon cancer, thyroid and pancreatic carcinomas have a mutation in one of the RAS genes. So, it tells the importance of the RAS gene and what happens if mutation occur in this particular gene. Now, this particular conversion of proto-oncogene to oncogene might occur by mutation of proto-oncogene like seen in RAS, or this might occur by rearrangement of genes in the chromosome, which moves the proto-oncogenes to a new location. Like c-MYC oncogene is found in human Burkitt lymphoma, and it caused because fragment of chromosome 8 moves to chromosome 2. It might also be caused by an increase in the number of copies of the normal oncogene, normal proto-oncogene, and it is seen in neuroblastoma when there is an abnormal increase in the number of copies of n-MYC gene. And the result of any of these genes is an altered form of proto-oncogenes, which will ultimately lead to uncontrolled division of the cell and formation of tumor. Now, these oncogenes, basically they are dominant mutation, and if a single copy is formed, then it will lead to production of the cancerous cells. And this particular type of mutation is also termed as gain of function mutation, because the cells, normal cells, do not have a specific function, but the mutant form gain specific function after the mutation. Now, the presence of oncogene in the germ cell line, either in egg or sperm, will lead to occurrence of this particular tumor or this particular cancer in the progeny of that particular individual. Coming to second class of the genes responsible for formation of tumor, we have tumor suppressor gene. As I have told already, they normally inhibit the cell growth and prevent tumor formation. Now, as the common sense suggests that if these particular genes are mutated, earlier they were inhibiting the cell growth, but if these are mutated, they will not be able to inhibit cell growth, and if that happens, then of course there will be uncontrolled cell growth and tumor formation. Now, mutation in these genes result in cells which are no longer able to show inhibition of cell growth and division, and the product of these genes may act either at the cell membrane or cytoplasm or in the nucleus. So there are a large number of sites where the product of these genes can act. Now mutation in these genes result in a loss of function, so they are usually recessive. In the proto-oncogenes we saw that there was a gain of function mutation, but herein what is occurring is that the protein is not able to inhibit, so there is loss of function mutation. So mostly these particular mutation are recessive. That means for the tumor to occur, there should be 2 mutated copies of the tumor suppressor gene. These genes are recessive genes. Unless both the copies are mutated, the tumor will not appear in the that particular tissue or organ. Now, in some cases, one of the mutation is already present in the germ cell. That means it has been passed from the parent to the offspring. And after a certain period of time, second mutation might also appear, because it is seen that the cells which already have one mutated copy, they are predisposed for the other copy to get mutated. Now, in the case of retinoblastoma, hereditary retinoblastoma, which is a serious cancer of retina which occurs in the early childhood, it is seen that if the individual receives one mutated copy from the parent, and those particular cells are predisposed to for their second copy to get mutated. About 90% of the offspring which receive one mutated copy, they develop a mutation in the second copy of the RB gene, and they show retinoblastoma early in life. Now, these retinoblasts basically are rapidly dividing cells, and because there are a large number of them, there is a high incidence of mutation in the second copy. Now, this particular disease afflicts only young children, because only the younger individuals have retinoblasts, which are the rapidly dividing cells, and this particular tumor appears by the age of 5. In adults, this mutation in the RB gene might lead to several other features. But not all the cases of retinoblastoma are hereditary. There might occur mutation in both the copies of RB genes in the somatic cells of the individual. Now, there are a large number of other tumors which are related with tumor suppressor genes, like familial adenomatous polyposis of the colon, then hereditary breast cancer, which results from mutation to both copies of BRCA2, and ovarian cancer. Then there is P53. DNA damage leads to induction of P53 tumor suppressor gene, which activate transcription of pro-apoptotic and cell cycle inhibitory gene. This P53 basically activates BCL2 family members, Puma and Noxa, and the cell which lack P53, they fail to undergo apoptosis. Therefore, they are able to divide in an uncontrollable manner. And this particular molecule also induces CDK inhibitor P21. Now, if there is a mutation in DNA repair enzyme, of course, there will be a severe problem to the cells. If DNA is getting mutated and it's not getting repaired, then we can assume what will happen to that particular cell. As is seen in the case of xeroderma pigmentosum, wherein the DNA repair gene is mutated, and these individuals show a high prevalence of skin cancer. Also in Bloom syndrome, where there is not a proper repairing of DNA, the chromosomes are not stable. Then coming to the last part, there are a large number of viruses which can cause tumors, like hepatitis B and C virus, papilloma viruses, herpes virus, retroviruses, and HIV also, which does not causes tumor directly, but it causes decline in the immunity of the organism, and it leads to tumors like lymphomas and Caspase sarcoma. Thank you.
Thank you, sir. Thank you so very much for giving us this session. Friends, we know that you have lots of questions in mind, and after watching the lectures on YouTube also, you might be thinking to get answers to your questions. So friends, you can write your questions at info.cec@nic.in. We'll try to give answers to your questions by next time Dr. Varunendra Singh Rawat visits our studio. Till then, keep watching us, keep writing us. We are going to meet again soon. Thank you, sir. Thank you so very much.